So, hello everyone. I'm a professor in the chemistry department, chemistry and biochemistry. I'm also an affiliate faculty in bioengineering and also an associate director of the California NanoSystems Institute at UCLA. I've been at UCLA for 17 years now. Prior to that, I got my degree in chemistry from Caltech with Nobel laureate Robert Grubbs, and also was a postdoc at the ETH in Zurich with Jeff Hubble, and in biomedical engineering. And I combined those two approaches here at UCLA. So my group creates new polymeric materials to address unmet needs in human health. And we have three main thrusts. So the first is, and our largest thrust, is in protein stabilization and delivery, where we develop new polymers to stabilize proteins to stressors such as heat and room temperature as well as pH. We also work in responsive medicines, where we have nanoparticles that deliver proteins directly where they're supposed to in the body. We also work on small molecule delivery, and we have an exciting new project on abuse deterrent technologies for opioid delivery. So I'd like to tell you today a little bit more about one of our platforms for protein stabilization, and that platform is Polyprotect. So I think most of you are familiar that protein therapeutics require special storage and handling due to their degradation. In fact, for the FDA-approved therapeutics, nearly 80% of them need to be refrigerated and stored. What you might not know, which is astounding, is that in 2020 it's predicted that the cold chain costs are going to be $18 billion. So this is a significant problem. And it's not only a cost problem, but it can be detrimental to the patients to have a drug that's not active. And so what we discovered is a polymer called Polyprotec. And this polymer is based on a natural stabilizer, which is trehalose. But in every stress that we've looked at, our polymers outperform the trehalose quite considerably. The polymers work to stabilize to heat, to lyophilization, to room temperature stabilization, refrigeration, and they work as an excipient, so added as well as conjugated. And if they're conjugated, they also increase the in vivo half-life, similar to the polymer polyethylene glycol, so they have both advantages. And I show here on the right a graph where we've heated the protein insulin that's used for diabetes. We heated that protein to 90 degrees Celsius, and you can see that we looked in vivo at a mouse model for an insulin tolerance test. We looked at the lowering of blood glucose levels, and what we found is, of course, the insulin is nearly inactive. It's less than 20% of its activity at lowering blood glucose levels, whereas if you add the polymer as an excipient, 80% of the activity is retained, whereas if you attach covalently two of the polymers to that protein, even after heating to 90 degrees Celsius, the insulin was 100% active. It's the same as the native protein. So we've been funded by the UC Innovation Award for this work, as well as the Physical Sciences PSEIF Award, and we have also been funded by the NIH and NSF. And what I'm asking for is we're very open to collaborations to move this research to first-in-human studies. And I have 3 posters here, and they're in the legacy room. And I encourage you to come and either talk to me or talk to my students. 2 of the posters are on the Polyprotect platform, and another poster is on small molecule drug delivery. Thank you very much for your time.